So good morning, everyone. My name is Tom Aherne, and I'd just like to thank everyone for the invitation to speak. So obviously, unlike my very illustrious co-panelists, I'm happy to declare that I'm not an expert or a serial trialist. However, I hope that I am proof that if I can perform a small trial, then I think that anyone really can. So as we all know, randomized trials are very high in the pyramid of evidence, and really they're the currency that we use in modern healthcare practice to guide our decision making with regard to patient care. And really, they're probably top of the pyramid, really, given that meta-analysis is often subject to a lot of heterogeneity. So NICE describe or define a trial as any study in which a number of similar people are randomly assigned to two or more groups to test a specific drug, treatment, or other intervention. So essentially, you're comparing two groups with a view to establishing which group responds better to treatment. And why are they important? I suppose they're important because they provide us with objective scientific observations. And when they're well designed, they can exclude bias and confounding and offer very reproducible and generalizable results so that your findings can be reproduced elsewhere. So you might say, what is bias? It's defined as any systematic aspect in study methodology which results in variation between the results of a study and the truth. So essentially, is there any way that the result that you found is not truly the way things are? And bias can be conscious or subconscious. And here you can see conscious bias, which obviously is unethical and best avoided. But a well thought out plan and protocol when you're initially designing your trial can make things very easy for you later on. So it's worth putting in a lot of effort at the start, and then the subsequent period of your trial life will be much easier, and hopefully you'll avoid bias. So what is confounding? Similarly, any variable that interferes with the relationship of the exposure and outcome, thus masking the true effect of the study variable. I thought this was a good example. So if you were studying ice cream sales and shark attacks, you'd probably find that they're both directly associated. However, when you bring temperature in, that probably has a significant bearing on both variables, therefore acts as a confounder. So therefore, when you're planning your trial, you need to try and take into account the effect of temperature to try and remove it as a variable when you're trying to assess the relationship of the two variables that you are assessing. So what are the strengths of trial data? They obviously compare groups. They minimize selection bias so that you're comparing relatively equal groups. They blind the patients and the assessors to remove the risk of performance bias and assessment bias. For example, you can imagine if a surgeon is performing a procedure and he knows it's part of a trial, they'll certainly endeavor to maximize their outcome, given that they know that they're under the microscope. All trials are prospective to outrule any recall bias, and a well-designed protocol, as I said, can account for confounding and ensure comparability. It also gives you the opportunity to stratify your groups to further mitigate against confounding, and ensures that you have statistical power to definitively confirm your results. And importantly, as a trainee who's working your way along, they're publishable, which is important, and it helps you progress through your career and gives you further opportunities to get involved in research. So obviously, not everyone does them. They can be difficult. And they can be tedious. You need to perform an initial power calculation. And often if you're looking for a rare outcome, you'll need a significant number of patients to ascertain statistical power. Therefore, do your power calculation early so you know exactly what you're dealing with and what numbers of patients you require to power your trial. They can take up a lot of resources, time, and they often are conducted across multiple sites. They do sometimes mask the effect of treatments. The conditions within a trial are often artificial, and patients are very closely followed up, which sometimes cannot reflect real life practice. There's also the issue of getting ethical approval, making sure that your trial is ethical. That process can be tedious and needs to be thought out effectively prior to commencement. And you need to ensure that you have equipoise, that there's a genuine fork in the road where you don't know whether the treatment you're instigating will make a difference. So why would you go to this trouble, really? I suppose if you're interested in genuinely changing clinical practice, trials provide robust evidence to support this. And this can potentially make you an influential figure in your field of expertise. As I said, you can exclude bias and confounding to make sure that your results are accurate, and you can publish your results and build your profile both locally and internationally. So getting started, the majority of the work for me, I think, is done before you actually start the trial. You need to set your goals and use the SMART acronym. I think your trial has to be very specific. It definitely has to be attainable and has to be time-based. When you're a trainee, you're usually in a position only for a year, maximum 2 years. So if you can't finish your trial in that time frame, I'm not sure is it worthwhile. So when you're starting out, have a very attainable goal. Make sure your hypothesis is simple, it's manageable, achievable, and bring an entrepreneurial spirit into it. Prepare to fail, but in failure, you'll learn an awful lot, so that when you come back to do your next trial or your next idea, you'll be far more effective, far more efficient in its conduct. Make sure it's a clinically relevant question, so that when you do eventually achieve results, that it will change practice. And try to avoid too much complexity in your initial efforts. Ensure that you have an adequate control group, and make sure, as I said, that you perform your power calculation early to ensure that you can realistically achieve the number of patients that you require. And probably most importantly, get help. There's lots of it available. Lots of people are enthusiastic about trials. So don't be afraid to ask and try and ascertain a mentor who will guide you through the process. So for me, when I was starting out, I found this document was the key to my whole understanding of trials. And essentially, it's the document which sets out its trials for dummies, essentially. And it provides you with a framework within which to design your trial, conduct your trial, and write up your results. They even provide you with pro forma diagrams that you can eventually insert into your document. So as I said, all the work is done early. You need to establish ethical approval. So try and form an informal relationship with someone on your ethical committee to get a sense of whether the trial that you're proposing will realistically get through the ethics process, because there is a lot of work involved with the ethical approval process for trials. And if it's unlikely to be successful, then I'm not sure is it worth persevering with. So try and sound somebody out as a sounding board whether you should pursue your particular hypothesis. The next step is to write your protocol, and essentially this is a map. And it's a map of where you're going and exactly how you plan to do it. Next step is to register your trial. It sounds onerous, but actually it only takes 30 to 40 minutes. It's free and you can do it on clinicaltrials.gov. Another useful tip is to identify protocols online from the major protocol journals like Trials or BMJ Open, and they'll give you another map of how to write your protocol. So naturally, the majority of your planning will be in the methods, and this is well described in the consort framework. So firstly, looking at participants, you need to identify who are your study population and how do they deal with your hypothesis. Establish criteria for inclusion and exclusion. If you have broad inclusion criteria, you'll be able to recruit more patients, your results will be more generalizable, but you'll be more prone to confounding. It's important that these patients are very accessible. Try to avoid doing studies on things that happen rarely. So try and identify patients in the outpatient clinic or high volume day case surgery. Offer a high turnover of patients, which potentially will help you generate your number for your trial. Identify where you're going to collect the data, whether you need to involve multiple sites, and all patients will require consent, and GDPR is becoming an increasingly bureaucratic issue with regard to ethics, so take that into account. So the interventions, when you're starting out in your trial career, it may be best to avoid invasive interventions, drugs, or interventions that are potentially harmful, because they involve a far more in-depth ethical approval process. And it may be better to start off with something less harmful, and to ensure that your ethical approval is granted and that you get your first trial over the line and get the experience that you require. Make sure that the intervention is performed uniformly, meaning that the intervention is performed in a systematic way and reproducibly. Recruit other sites if you need a significant amount of numbers to power your trial. And ensure that there isn't a significant variation from current practice. And what I mean by that is try and add little tweaks to current practice when you're trying to establish your hypothesis, rather than trying to reinvent the wheel. So when you're looking at your outcomes, these should be easily measured, they should be reproducible, and you need to define them well in your protocol. Try to use validated tools that have been used in other studies that look at similar hypotheses. Try to avoid self-generated questionnaires and tools like that. Search for common outcomes, because that will mean that it's easier to generate a number of patients for your trial, and limit the number of outcomes to reduce complexity. So when you're looking at your sample size, examine previous evidence and try and get an assessment of the effects of previous trials. As I mentioned, rare events need greater numbers to achieve significance, and there are a number of easy online tools which can be used to generate an estimate of a power calculation, which can guide you. If there's also plenty of tutorials online about how to statistically generate a power calculation. So try and do this early so that you know exactly what you need to do and whether your trial is indeed achievable. The next step is randomization and allocation concealment. And this should be computer generated using random block sizes. Essentially, this can be done on Excel. And again, there's excellent tutorials on YouTube that you can use to teach yourself how to do this. Therefore, you should obtain a third party who's independent of your trial to generate the sequence and then transcribe it onto opaque envelopes, which is the easiest way to ensure allocation concealment. There are online tools to do this, such as sealed envelope, but they're associated with a significant cost, and with a little bit of self-directed teaching, you can do it yourself. Consider minimization if your trial is small. What minimization is a method of randomization that allocates subjects to a treatment group that best maintains the balance of prognostic factors. Essentially what this does, it ensures that individual patient variables are equally distributed between both groups to limit the effects of confounding. And you may well require a statistician to perform this, so take it into consideration. So implementation of your trial and blinding. It's important in your protocol to establish who's doing the randomization, who is recruiting, who is assigned the patients to intervention, who are doing the interventions, and who is blinded. And really, again, with your early trial experience, you need to assess what is feasible. Ideally, you would have an independent third party doing all these who are blinded, but that's not always feasible. Therefore, try and make your trial as scientific as possible, but it also needs to be achievable. So recruitment, you certainly need to involve a team and try and engage your colleagues. So you need to make it easy for them to be part of your trial and also worthwhile. Everybody's interested in being part of a trial or having a publication of a trial on their CV. Therefore, it is easy to convince people when you make the process easy for them. Try to ensure that you have easy access to recruits. As I said, target high volume areas of practice, such as outpatients or surgical day wards. Have packs made up of questionnaires and consents so that they can be left on a ward for your colleagues to use. All patients will obviously require a consent. And it's best to try to transcribe your data that you collect in real time and design a user-friendly database to store your information, because it can be challenging when you when you're running a trial off sheets of paper. So follow-up, again, for your first trial, try and make the follow-up time short. The longer the follow-up, the more times you need to see them, the more complexity is, and the higher the rate of attrition bias or your loss to follow-up. If it's possible to design a trial that has follow-up all performed within 1 day, that would be ideal. If not, try to assess whether you can follow patients up over the telephone, which is easier. Try not to rely on patients or staff, because oftentimes this will lead to loss to follow-up. And try to avoid patient notes, because often they can be very difficult to attain once the patient has been discharged from hospital. So the key points that I hope I can pass on are, it's all about planning. So spend most of your time planning your trial. Start with something very easy. Make your mistakes there. If it fails, just get back on the horse, because the next time around it will be a lot easier and a lot more efficient. Try and limit the variables and the moving pieces in your early trials. Engage colleagues, and certainly there's plenty of help available around, and there's lots of people who are interested in being involved. Establish mentors who will guide you through the process, and certainly this engagement will open up further opportunities for you, and further research opportunities. The last thing I think I would say is try to never pass an opportunity. I know everybody online is extremely busy and has a lot going on, but try not to pass an opportunity when it comes your way, even if you are busy, because it's rare that you regret it when you come to the end of the process. So thank you. And if anyone has any questions or wants any assistance, I'm happy to take emails at any stage. Thank you.